Okay, so hello everyone, it's igcse.science.notes with Taqwa team. And in this video, we will be doing Biology Unit 2 repetitive and most important questions. So let's get started without wasting further time. Starting with the first question, it's one of the most important and repetitive questions ever. Here, they tell you describe how the rough endoplasmic reticulum and Golgi apparatus are involved in the processing and secretion of proteins. I want you to know that this question might come in another way. They might tell you, is the rough endoplasmic reticulum and Golgi apparatus involved in processing and secretion of proteins? It might be MCQ question. It might be, what is rough endoplasmic reticulum and Golgi apparatus involved in? So you need to say secretion and processing of proteins. Okay, so let's know how is it involved. First of all, let's color the rough endoplasmic reticulum in RER and know that it has another name called RER. Okay, so it's folded into tertiary structure in the rough endoplasmic reticulum, and proteins are packed into vesicles at the end of rough endoplasmic reticulum. That's how the rough endoplasmic reticulum is involved. Protein is folded into tertiary structure in the rough endoplasmic reticulum, and they are packed into vesicles at the end of the rough endoplasmic reticulum. For the Golgi apparatus, the proteins are modified in it, and the modified protein is packed into secretory vesicles by Golgi apparatus. What also happens is that the glycosides or carbohydrates are added, and so the proteins leave the cell by exocytosis. Okay, that's great. Second question, which comes a lot, and inshallah we'll be doing a separate video about mitosis and meiosis because it's very important. This question tells us, describe the changes that occur in the prophase. What is the prophase? Prophase is basically when the nucleolus disappears and the nuclear membrane breaks down, so the centrioles move to opposite poles, and so the spindle fiber forms, then the chromosomes become visible, and as pair of sister chromatids, the centromes hold chromatids together. Again, because it's important, let's repeat. Describe the changes that occur in the prophase. The nucleolus disappears, the nuclear membrane breaks down, the centrioles move to opposite poles, the spindle fiber form, chromosomes become visible as pair of sister chromatids, centriomes hold chromatids together. Okay, that's great. I told you Golgi apparatus and RER comes lots of questions about. Here's another question that mentions Golgi apparatus. Describe the differences between the structures of mitochondria and Golgi apparatus. Again, let's color mitochondria in a color. I think mitochondria looks like blue or somehow purple, and Golgi apparatus that has the color red. Okay, so the mitochondria has double membrane, while the Golgi apparatus has single membrane. Cristae are present inside the mitochondria, while there is no in the Golgi apparatus. The Golgi apparatus is made of cristae, while mitochondria is rod-shaped. These are the differences between the structure of the mitochondria. As you know, mitochondria is the powerhouse of energy in the cell, and the Golgi apparatus. So the Golgi apparatus again is made of cristae, does not have cristae, and has single membrane, while the mitochondria has double membrane, cristae are present, and it's rod-shaped. Okay, this is somehow Unit 3 type of question, but we will also be explaining over here. Describe how to prepare a root tip squash so that chromosomes can be seen. First of all, we remove the last 7 millimeters of root tips. Then use the hydrochloric acid to separate the cells. We then after add the toluidine blue. Heat the slide to intensify the color, and then place the root tips on microscope slide, cover and squash to separate the cells. That's great. Let's now cover the 5th and 6th and 7th most important questions you'll ever see in the Unit 2. Question number 5. Let's change the color, choose this one. Great. Give the differences between a human sperm cell and a human egg cell. The human sperm cell has a flagellum, is streamlined, and has acrosome, while the human egg cell has zona pellucida and a cortical granules. Okay, question number 6. Looks like it's the same color. Give the similarities between a human sperm cell and a human egg cell. They both have nuclear pores, both are haploid, and both produced by meiosis. So again, let's have this color. The differences are these. The similarities are these. I'll be posting this PDF inshallah on the Telegram channel, so do not worry. And also the WhatsApp channel on Taqwa Team and on Telegram on igcse.science.notes.z. Okay, great. Let's complete. Explain how mitosis causes the genetic variation in gametes. There will be another question which says, what causes genetic variation in the gametes? We say mitosis. So how is mitosis causing genetic variation in the gametes? Basically, independent assortment, and the four daughter cells have different combination of chromosomes, also the crossing over, and that the DNA are swapped over. So this is how the mitosis causes genetic variation in the gametes. It's basically that the four daughter cells have different combination of chromosomes, and so there is independent assortment, and the crossing over and DNA that are swapped over also causes the genetic variation, which is also caused by the mitosis. Let's do question 8 and 9. Now, question 8 asks us to describe the function of the following structures that are found in a human egg cell. So let's choose another color again. Basically, the lipid droplets and the cortical granules are found in the human egg cell. But what are their functions? The lipid droplets is there to provide a store of energy, while the cortical granules is there in the human egg cell to secrete, like it's there for secretory vesicle to move to egg cell membrane so that the enzyme released and the zona pellucida hardens, which presents the polyspermy. Again, what are the functions of the lipid droplets? It provides a store of energy with cortical granules, secretory vesicles, move to egg cell membrane, enzyme release, zona pellucida hardens, which prevents the polyspermy. Okay, describe and explain ways in which a human sperm cell is specialized for its function. We said, first of all, that the sperm cell has a flagellum, is streamlined, has an acrosome, has nuclear pores, is a haploid, and is produced by meiosis. So how are these characteristics, or how is it itself specialized for its function? So basically it has flagellum, as we said, it has a haploid nucleus, it has chromosome, right? It allows restoration of diploid chromosomes at fertilization since it's a haploid nucleus. Also, it has a hydrodynamic shape. It has reduced resistance. It's involved in the breakdown of the zona pellucida. Has large number of mitochondria, which helps it move by the flagellum to supply ATP. Large number of mitochondria to supply the ATP, so it has a good amount of energy, and for propulsion. This is how the sperm cell is specialized. Again, how is the sperm cell specialized? In case someone just joined, basically it has a hydrodynamic shape, reduced resistance, acrosome containing acrosin involved in breakdown of the zona pellucida, haploid nucleus, allow restriction of diploid chromosome of fertilization, large number of mitochondria, supply ATP, flagellum present for propulsion. That's great. Okay. I want you to explain how acrosin is released from the acrosome. So basically here, one of the really good specialization for the function is that the acrosome contains acrosin. Not only that it has acrosome, that it contains acrosin. So why is it important that there is acrosome, right? And how is it released? So basically the receptors come in contact with the zona pellucida. So the acrosome swells and then the vesicle fuse with the sperm cell membrane by excitosis. So the acrosome is released from the chromosome. Again, how is it the acrosome released from the acrosome, which is very important. Basically, the receptors come in contact with the zona pellucida, which is on the egg cell. The acrosome swells, vesicle fuse with sperm cell membrane, exocytosis. Right. In the cell membranes, we saw also that the mitochondria is important, is there a lot for supply of ATP, right? But what other function is it there, or why is it important? Like, why do we need energy? Why is it like energy is needed? To move the flagellum, to allow the sperm to swim toward the egg. the mitochondria provides ATP, so there's carrying out of aerobic respiration inside the cell. Okay, let's change the color. Looks a bit weird. Purple is there. Orange. Okay. Explain the importance of meiosis in the formation of human sperm and egg cell. We said that the human sperm and the egg cell both are haploid. And so we said both are made of meiosis, right? So why is the meiosis important? Because it has the chromosome number. Like in human being, there are 46. So in human sperm cell and egg cell, there is 23. Half. So that at fertilization, the full complement of chromosome is restored. Chromosome from the parent and chromosome from the parent, 23 plus 23 makes 46. Allow genetic variation in gametes, which we said earlier, through independent assortment, which we said also earlier, through crossing over, which we also said earlier. Make sure that it's very important to memorize these things. Question number 13 is very small, but it comes a lot. Describe the process of double fertilization at flowering plants. Number 1, one male nucleus fertilizes the egg cell nucleus. Number 2, one nucleus fertilizes the two polar nuclei. Again, how is the double fertilization in flowering plant occur? What's the process? One male nucleus fertilizes the egg cell nucleus. One nucleus fertilizes the two polar nuclei. Okay. Because it's called double fertilization, this is how we can memorize it. Let's go into question number 14. Question number 13 also talks about flowering plants and pollen tubes. So you need to know that there is a function of the pollen tube in fertilization in flowering plants. So let's choose a green color because we're talking about plants. How they're important in fertilization? Basically, it forms a pathway through the style, grows towards the ovary, digestive enzymes, transport generative nucleus, and then fuses with embryo sac. Okay, describe how cells become specialized. You know, there are some cells that are not specialized at all, they're just there, right? So how they become specialized? Through stimulus. It activates some genes, then the transcription at active genes, then the mRNA is translated to produce the protein, and then this protein is permanently modified by the cell. Explain what is the term meant by the polygenic inheritance. What is polygenic inheritance? Polygenic inheritance, more than one gene for a single characteristic at different loci. Loci means location. Comes from the word locus. Locus is location. So what is polygenic inheritance? More than one gene for a single characteristics at different loci. Loci is location. Describe the structure of cellulose microfibril. Microfibril is stated in a lot of questions. So what is the structure of the cellulose microfibril? So basically, as normal cellulose, it's made of beta glucose. And as normal cellulose, it's joined by condensation reaction. And as normal cellulose, it has 1,4-glycosidic bond. And as normal cellulose, they are held together by hydrogen bonds. And again, as a normal cellulose, they are alternate glucose molecule are inversed. And again, as normal cellulose, they're made of straight chain. The only one thing that has to do exactly with microfibril is that the microfibril is composed of many cellulose molecules. You just need to say that. Okay, so again, what is a cellulose? Let's just remove what we've done and cover this word. Describe the structure of the cellulose. We're going to say that it contains beta glucose. The word beta shows that it's made of an alternate glucose molecules and they are inversed, and that cellulose, we need to know from the biological molecules earlier in unit 1 that they are joined by condensation reactions, and it's 1,4-glycosidic bonds, and it's straight chain, and the cellulose molecules are put together by hydrogen bonds. The only thing that has to do with microfibril is that it's composed of many cellulose molecules. Okay. Explain how the structure of cells in the tissue enables it to be strong and flexible. We've just said over here that it's straight chain, right? So basically, the cellulose is basically also used in that. So, whatever about the cellulose, explain how the structure of cell walls in this tissue enables it to be strong and flexible. Basically, cellulose microfibrils. The cellulose microfibrils, as we said, it's straight, right? So, it's very important in that because it's like held straight, no branch, no anything from that. And so, it enables it to be strong and flexible. Also, microfibrils parallel to one another a layer for flexibility, the mesh of microfibril and secondary cell wall for strength, cellulose microfibrils embedded in pectin. Again, let's just give a name for this. We said here, explain how the structure of cell walls in the tissue enables it to be strong and flexible. So we're talking about cell walls, right? But what in cell walls make it strong and flexible? The cellulose. Cellulose only? No, the cellulose microfibrils. So why is the cellulose microfibrils important? Because the microfibrils are parallel to one another in layer of flexibility, and that the mesh of microfibril and secondary cell wall is for strength. Also that the cellulose microfibrils are embedded in pectin. Okay, this is also somehow a question, is Unit 1 question. Compare the structure of cellulose with that of starch. So we said earlier that the cellulose is made of beta glucose, right? But we need to know that starch is made of alpha glucose. Starch is composed of more than one type, basically amylose and amylopectin, while cellulose is made only of beta glucose. Cellulose is straight, starch is branched. They're both made of condensation reaction. They both have 1,4 glycosidic bond, while starch has also 1,6 glycosidic bond. That's why it's branched. And they're both made of glucose. Starch is made of alpha glucose, cellulose is made of beta glucose. Okay, suggest why calcium ions affect the tensile strength of plants. Because it has calcium pectate, metal lamella, holding together microfibrils and cell walls, and that the fibers are stronger if microfibrils held together more strongly. Again, some plants questions. It was all about plants, even though if it wasn't direct. What is the structure of xylem? The xylem lacks cytoplasm. It has thickened cell wall, presence of lignin, cells are joined end to end. Describe how xylem is adapted to its function. Yes, it has no cytoplasm, it has thickened cell wall, it has presence of lignin, and the cells are joined end to end. But how is that good to its function? It has two functions, support and water transport. It's good for water transport as it has hollow tubes, vertical movement of water, waterproof material, less water lost, and pores to allow sideways movement of water. It's good for support as lignin for strength, rings for flexibility. Okay. Compare the function of xylem vessels with the sclerenchyma fibers. So basically, both are used for structural support, and only xylem transport water, and only sclerenchyma fibers transport minerals. Because xylem transport water, there is a mistake over here, guys. So there is a mistake here because xylem only transports water, right? Okay. Water and ions and etc., while sclerenchyma fibers does not transport water. Okay. Let's describe an experiment to find the optimum concentration of nitrate needed by a plant. Use clones, plants of same age, use 10 different nitrate concentrations, keep temperature and light intensity constant, measure the mass as a measure of growth, allow 2 weeks as time for growth, replicate at each concentration. Again, I would like to leave you a minute or something to read it because this is important to know by your own. Okay. So basically, the as we said, we need to describe an experiment to find the optimal concentration of nitrate needed by plant. We use clones, plant of same age. We use 10 different nitrate concentration. We keep the temperature and light intensity constant. We measure the mass as a measure of growth and allow 2 weeks for growth and replicate at each concentration. Question number 25. Explain what is meant by a double-blind trial. One set of patient given the drug and one set a placebo. Neither the patient nor the doctor knows if the treatment contained drugs or not. This is what we call by the double-blind trial, and I believe it has somewhat to do with sociology. Anyways, explain what is done in the phase 1 and phase 2 in the drug testing. Basically, the phase 1, it's tested on small number of healthy individuals with concentration of low concentration of drug. In phase 2, it's tested on small number of patients, monitor effectiveness of treatment. Explain why placebos are used to test the efficacy of new drug treatments in active substance comparison with actual drug, and people believe that they improve. Suggest how a valid investigation can be carried out to produce reliable data to compare the tensile strength of two types of plant fibers of same length, temperature controlled using air conditioner, adding masses until fiber breaks, and repeat and find mean. Also remove the anomalous and repeat. Anomalous means like if there's every results are like 10 and then there's an anomalous result of 100. Abnormal one. Force over cross-sectional areas, tensile strength, and then wear goggles to protect your eyes. Describe an experiment to compare the antimicrobial properties of two species of plant. Extracts made using fruits of same mass. Extract made using same solvent, which is ethanol. Same volume of extract tested on bacteria. Same species of bacteria used. Incubated at 27 Celsius for 48 hours. Zones of inhibition measured to calculate the mean. Explain how breeding programs can help to conserve the genetic diversity of animals. Use of DNA profiles to select mates, exchange animals between zoos, reduced interbreeding, prevention of genetic drift, the introduction of alleles from other populations. Suggest how the captive breeding and reintroduction programs can be used to conserve the animals. So basically, the increase in population size, the increase in genetic diversity, the exchange of animals between the zoos, the use of DNA profiling to select the mates, the reduced food intake to encourage hunting, the choice of the national park for a reintroduction, and the protection from predators. Describe what seed banks will do to the seeds to ensure long-term conservation of the seeds. This is very important. Number 1, they use X-rays to select seed with a living embryo. Clean the seeds, dry the seeds, store at low temperature, regular testing for viability, and if viability is less than 75%, select the fresh seeds for storage. Question number 33 asks us to describe how biodiversity can be measured. Basically, by counting the number of species in a known area of land, a number of different alleles in a species. Describe how seed banks help conserve the genetic diversity of endangered species. Seeds harvested from different plants of same species. Seeds collected from plants growing in different locations. Large gene pool. Why seeds are... Sorry guys, I think there was like an internet connection or something. So why seeds are dyed? To prevent enzyme activity, to prevent germination of seeds, to prevent the microbial growth decomposition of seeds, and drying reduces the damage when seeds are frozen. Again, why the seeds are dried? To prevent the enzyme activity, to prevent the germination of seeds, to prevent the microbial growth and decomposition of seeds, and it reduces the damage when seeds are frozen. Here also we said that one of the most important things to do to ensure long-term conservation of the seeds is to dry the seeds. These are some experiments that we will be mostly covering also in Unit 3. So I just want to tell you that thank you for a near tutory. It was such a pleasure finding these notes. Thank you so much guys for watching this video, and we will have just a 5 minutes break and then go back, finish this video by doing the important keywords and definitions, inshallah.